Today's seminar is about Progeria Syndrome or Hutchinson-Gilford Progeria Syndrome, which is prepared by me, Lava Abdulwahed, and supervised by Dr. Abdulrahman Khalil. Table of content consists of introduction and definition, epidemiology, genetic cause and how progeria causes disease, symptoms, diagnosis, treatment and management, research and future hopes, and references. Introduction and definition. We start by progeria. Progeria or Hutchinson-Gilford Progeria Syndrome is a rare genetic disorder that causes premature aging in children. The name comes from the Greek pro means before and geras means old age, so it literally means aging too early. Babies born with progeria usually look completely healthy at birth, and there are no immediate signs that anything is wrong. However, within the first few years of life, parents and doctors start noticing that the child is not growing properly. They are smaller and lighter than normal. So they start losing their hair, their skin becomes thin, and they develop features that make them look much more older than they actually are. Despite these physical changes, their intelligence, personality, and emotions are completely normal. So they go to school, play, and learn like any other child. So progeria is not just about appearance, it's a disease that affects nearly every cell in the body, except the brain. For the epidemiology, progeria, as we know, is extremely rare and one of the rarest disease known. According to the Progeria Research Foundation, it affects about 1 in every 20 million live births worldwide, and there are currently around 350 to 400 known cases across the globe. It affects both boys and girls equally and occurs in all races and nationalities. So one important point is that progeria is not inherited from parents, so it's not genetically inherited disease from parents. It usually happens due to a new mutation in the child's gene, what science call a de novo mutation. So it means even if both parents are perfectly healthy, they can still have a child with progeria. And the first visible symptoms usually appear about 6 and 12 months of age when parents notice growth failure and early hair loss. Genetic cause and how progeria causes disease. So the root cause of progeria or the causing of the progeria lies in a single gene called LMNA, which is located on chromosome 1. So this gene is responsible for producing a protein called lamin, and this lamin plays a key role in supporting the nuclear envelope or nuclear membrane, which is like the protective shell around the cell's nucleus or the control center that holds our whole DNA. In progeria, a tiny change or a small change in the LMNA gene occurs, a single letter switch in the DNA sequence, which causes the cell to make a defective version of the lamin A called progerin. Normally, lamin A helps maintain the structure and strength of the nucleus, but progerin behaves differently, so it sticks to the nuclear membrane and builds up inside the cell, damaging it over time. This single mutation makes a chain reaction that leads to a rapid cellular aging, tissue damage, and physical symptoms we see in children with progeria. It's amazing to realize that one small change in our genetic code can lead to such a dramatic effect on the entire body. So the symptoms are, it can be two-part, physical feature and other features. For the physical feature, it happens in growth failure, hair loss, thin or wrinkled skin, a small face with a big nose and large eyes as is shown in the picture, prominent vein on the scalp, loss of body fat and muscle, joint stiffness and weak bone. Other features can be normal intelligence, emotional development, or no developmental delay in thinking or speech. As we said before, it doesn't affect brain. And lastly, main medical problem is the heart disease and stroke at a young age. Most deaths of this disease is by stroke. For the diagnosis and the treatment, so diagnosing progeria is usually based on physical signs and growth pattern. Doctors can often suspect by simply observing the character appearance, for example, their small face, hair loss, and growth delay. However, to confirm the disease, a genetic test is done to check for the specific mutation in the LMNA gene on chromosome 1. So other additional tests may include, for example, echocardiogram, bone test density scan, or blood test. For example, echocardiogram for the heart health, bone density to look for osteoporosis in the bone of the individual, or blood test for cholesterol and sugar level. So once the diagnosis is done, families mostly are referred to genetic counseling, so they receive information about the condition and emotional support to cope with it. Early diagnosis is so important because it allows doctor to begin management and treatment early. And unfortunately, there is no complete cure for progeria yet, but major progress has been made. The most important advancement is a drug called lanofarnib, which is sold under the brand name Zocin VY. This drug was approved by the US in 2020, which the administration is called FDA, Food and Drug Administration. They make it globally the first global approved treatment for progeria. So this drug is the first one that is approved generally or globally. Lanofarnib works by blocking a chemical process called farnesylation, which helps the progerin attach to the nuclear membrane. By stopping this process, lanofarnib improves the shape of the nucleus, helps cells live longer, and extends the lifespan of children with progeria by several years. In addition to medication, a supportive care plays a key role. For example, regular heart checkup and blood vessel checkup, balanced diet and physical therapy, dental and orthopedic care, and lastly, emotional and social support for families. Without treatment, most children live until their early teens, 13 to 15 years. With new treatments like lanofarnib, some live into their 20s, and the main cause of death is heart disease or stroke, similar to elderly adults. And for the research and future hopes, research on progeria is very active and full of hopes. Scientists are trying to target the root or the main problem or the main cause of the LMNA gene mutation and stop this mutation and stop the production of progerin together. So several advanced approaches are being studied. For example, gene therapy to cure the LMNA mutation, CRISPR genome editing. So this experimental studies in animals show promise, or in here they aim to fix the DNA error directly, or they try to fix this DNA problem. And finally, the third one, antisense oligonucleotide therapy, which uses short strands of RNA to block progerin formation, and senolytic drugs, which helps remove old and damaged cells. And also combination therapy is used using lanofarnib with other medicines to stop the growth of this disease. And these are the references of my seminar. Thank you for listening.